Hello, everyone. Welcome to today's live broadcast, Invisible Influence. I'm Robert Castellanos of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is a leading scientific social networking website and producer of educational virtual events and webinars, contributing to the advancement of science through content sharing capabilities. LabRoots is a powerful advocate in amplifying global networks and communities. For more information, please visit labroots.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problems through the green Q&A button lower left. This is an educational webinar and thus offers free continuing educational credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. I would like to now introduce today's speaker, Dr. Jack Gilbert. Professor Jack Gilbert earned his PhD from the Unilever and Nottingham University, UK in 2002 and received his postdoctoral training at Queen's University, Canada. He has then returned to the UK in 2005 to Plymouth Marine Laboratory as a senior scientist until his move to Argonne National Laboratory and the University of Chicago in 2010. Currently, Professor Gilbert is director of the Microbiome Center and professor of surgery at the University of Chicago. He has authored more than 200 peer-reviewed publications and book chapters on metagenomics and approaches to ecosystem ecology. Finally, he's a founder, editor-in-chief of M-Systems Journal. I will now turn it over to Dr. Gilbert for his presentation.
Right. Okay, I hope everybody can hear me. It's a great honor to be here. I apologize for the state I look like I'm in bed. I actually am, but not actually in bed. I'm just in the spare room of my house. However, it's my great honor to be able to present to you all. I'm going to be talking today about how the microbiome in individuals and people can affect outcomes in our patients, and more specifically, how we are using the microbiome in order to understand the way in which the body responds to treatment, and how we can understand the specific implications of lifestyle, of immune variants, and even of surgical practice upon outcomes in patients. So I'm going to specifically also talk about the indoor environment. I do run the Microbiome Center at the University of Chicago, the Marine Biological Laboratory at Woods Hole, and the Argonne National Laboratory. This is a multidisciplinary effort with more than 60 faculty, over 30 departments focused in this area. So, start out with a little story. I have a variant, a gene variant that gives me a predisposition towards the development of Alzheimer's. So I have a likelihood of more than 25 times over normal of developing Alzheimer's disease. I got this from my mother. She has the same variant, but she has a double mutant of this variant, which gives her a heightened level. But even that doesn't mean she'll actually develop Alzheimer's disease. It's far more likely that the environment in which she lives, the environment in which I live, might select for my predisposition and leave me open to a more specific disease phenotype. So the reason for this is, and one of the key areas of environmental interaction is the human microbiome. It's the some 30 to 50 trillion bacterial cells present in and around your body. There's around 1.3 times the number of bacterial cells in your body as there are human. That 10-to-1 ratio that keeps on being bandied about is actually a back of the envelope calculation error. But this microbiome that we find in our body actually comes from the environment and is shaped by how we interact with the environment, both through our diet, our lifestyle, and various other phenotypes. And so what we are interested in doing is understanding how the microbiome could be playing a role in developing our predisposition and developing the actual development of specific conditions. We get our microbiome predominantly from our mother in the initial phases. We see this at birth with a rapid inoculation through vaginal delivery of the vaginal microbiome. So here we have, for example, it's a PCA plot. Here we can see each dot is a microbiome sample representing the relative abundance of some 1,000 species in each dot. And what we're seeing is that samples that come from the mother's vagina versus the babies that were born by vaginal delivery have a more similar microbiome. Those red and pink dots are closer together. Whereas children that were born via C-section, their microbiome is much more similar to the mother's skin. So they develop a microbiome that's more akin to the skin environment which they first interact with. You can see the mother's mouth at the top there in yellow. It's very different from both of these environments. So this is the starting culture, if you will. This is the sourdough bread or the craft beer that you would normally acquire early in life. And we think this is incredibly important. Children born via C-section have a heightened risk of developing atopy, so allergies, asthma, those kind of things, and skin conditions. And they also have a heightened risk of developing obesity and diabetes, as well as some neurological conditions. Many of these conditions have been likened or linked or associated with changes in the microbiome. And therefore, it's not too much of a leap of faith to understand that disrupting this early phase of microbial development, i.e. from a predominantly vaginal microbiome start to a predominantly skin microbiome start, could have an impact upon the developmental trajectory of a child, predisposing them to some conditions. Again, it doesn't necessarily mean they will get those conditions. Many children are born via C-section and go on to live totally normal, healthy lives. But there's an increased probability of a potential threat to their long-term chronic health. The fact that we get, after we're born, we interact with the environment is obviously axiomatic. But this leads to a unique microbial community. All of us have a unique microbiome. In fact, I can identify each one of you in the audience or myself or my family based on the microbiome signature. And I could use that in clinical, I could use that in criminal investigations, not just clinical investigations, but criminal ones. There's some work we're doing currently with the National Institutes of Justice, which points in this direction. We might bring that up later, but even identical twins have a unique microbiome. They obviously acquire the same microbiome from their mothers at birth, but then as they grow up, one may be licked by a dog and the other one may be, likes to play in the dirt a bit more. And those microbial exposures shape the types of bacteria that they acquire. Now, whether the microbiome they acquire shapes their community by directly allowing organisms to colonize their body, or whether the experiences they have with the world shape their immune system, which it directly shapes the microbiome through selective acquisition, retention, and killing, we don't know. We think it's a bit of both. So we're working on the premise right now that increased microbial exposure with the environment is incredibly important, and I'll talk about that in a minute. We do know that we are currently eroding our microbiome, leading to a reduction in the diversity in our Western world, and also a reduction in the types of bacteria that we find on our bodies. And this has led to a significant increase or a surge in our attempt to understand how the microbiome and environmental exposure may play a role in certain conditions. There are currently 399 ongoing clinical trials many of those using a treatment called fecal microbiome transplant, where we take the microbiome of a healthy person and transplant it into an unhealthy person in order to improve their gastrointestinal health. And we can see that the microbiome is now being implicated in many individual conditions. And we're also finding that many individual treatments, like, for example, artificial sweeteners, or even situations like antibacterial soaps, triclosan, etc., can have an effect on the microbiome. In fact, we just wrote a editorial for Science on this, and then 1 week later, the FDA banned it after exploring triclosan for about 40 years. So I'm not saying that our editorial is directly linked to the FDA's decision, but you can ever hope. So how does the microbiome influence? Well, these are three nuggets we like to tell in our community. One, work we've done with the Department of Defense has identified that the microbiome alters the skin chemistry through metabolizing sweat and metabolizing oils to produce aromatic carbon, that influence the attractiveness of an individual site on the body to mosquitoes. Mosquitoes sense you in the area by honing in on your carbon dioxide cloud, but then when they get down to the body, they decide which site to bite by actually honing in on the unique series of hydrocarbons, aromatic carbons, that are being released from the site. So there are some mosquitoes that are predilect biting the wrist, and there are specific types of bacteria living on my wrist that metabolize sweat in a specific way, releasing certain types of aromatics, whereas the bacteria on my ankle are very different, and they release other types of aromatics, and different types of mosquitoes are more attracted to that site. Being able to disrupt that signal could lead to a significant reduction in the transmission of diseases spawned by biting insects. Tylenol is only toxic to your liver if you have certain types of microbes in your gut. There's a microbial interaction between the chemicals in Tylenol and the metabolism products, which leads to an interaction in the liver, causing liver toxicity. So we could theoretically screen people's stool to look for certain types of bacteria which would predispose someone to being to having a heightened risk of liver toxicity due to acetaminophen treatment. We also know there's a picture of two flies humping, which is a strange thing to have in any slide, but in this instance, we know that fruit flies can change their behavior by having their microbiome altered. In fact, I can take the microbiome of one fruit fly, transfer it into the gut of another fruit fly, and then alter the fruit fly that the first fruit fly fancies to the second fruit fly. That's a strange tongue twister, but the general principle is that the microbiome in the first fruit fly is actually affecting its mate preference. And therefore, by transferring it into the second fruit fly, I can alter that fruit fly's mate preference to that of the first fruit fly. So I can change the sexual behavior of an animal by altering the bacteria present in its body. You might think that the sexual behavior in an animal is always predicated by its genome, but this proves not to be the case. So we're very interested in getting the microbiome into your medical records. And in fact, there's now clinical evidence to suggest that the microbiome and microbial therapies could be extremely important. The most widely cited of these is the use of curing C. diff with a fecal microbiome transplant. Here we can see typical vancomycin or vancomycin with bowel lavage treatment for C. diff has around a 30% success rate for people with chronic infection and who are not responding to treatment. Whereas an infusion of donor feces, a fecal microbiome transplant, can lead up to a 94% recovery rate. In this groundbreaking study, the study was actually cancelled early because it was viewed as being so successful it would have been unethical to continue to treat the control population. So what do we think about the microbiome and how do we see it as being important? Well, we've gone from living outdoors with a rich microbial exposure to living indoors with a very limited microbial exposure, which we've eradicated because we use antibiotics that kill the good bacteria along with the bad ones, as well as that triclosan. And we think this is reducing the microbial diversity of our bodies. So we're leaving ourselves open to the development of allergies, atopy, and various other things. We think that by actually increasing the microbial diversity or adding certain very specific types of bacteria back into our body, we may be able to protect our children and adults from some of the allergens which are leading to some of the chronic illnesses we see in the Western world today. In a paper from the New England Journal of Medicine that we published not more than 2 or 3 weeks ago now, we demonstrated that dust from the farms such as an Amish farm can have a protective effect upon the bacteria protective effect upon certain mice strains that are predisposed towards the development of asthma. So that by taking the dust and their bacteria and allowing these mice to experience that dust, we can protect them from developing asthma. Now, this is in contrast to the dust from a home of a Hutterite, which is a different type of religious sect, where the children in that farm are not allowed on the farm because the farm is a very mechanical, very different environment from that of an Amish farm. Now, while the Hutterites live a very similar home life to the Amish, they don't live a very similar farm life. And we think this reduces the types of bacteria that enter into the home environment. And in fact, where the Amish have around 1% asthma in their population, the Hutterites have more than 25 to 30% asthma in their population. So there's a big disparate gap between people living in continuous contact with the animals and people who are disrupted from that animal interaction. Remember, the Amish children are playing in the farm all the time. The home is on a farm, whereas the Hutterite children don't ever get near the farm. We also know that we can alter the microbiome of a mouse and affect its ability to be sensitive to particular allergens. So in this peanut allergy study up here, the peanut-specific IgE response, we can see that a specific pathogen-free mouse, standard mouse model, has a very low response to peanut-specific IgE, to the allergen, causing a low response of the IgE uptake in its serum. Whereas a germ-free animal, the GF in the red column, has a significantly elevated degree of response to the peanut allergen, causing an order of magnitude higher increase in response in the variable. If we reconventionalize those germ-free mice, that response goes away. They're no longer responding to the allergen. If we add in Bacillus uniformis, we actually don't see the protective effect. This type of bacteria doesn't offer a protection to a germ-free animal. If we take a germ-free animal and we just add clostridial organisms, particularly Clostridial cluster 14A, we see a significant reduction in the response of that animal to a peanut allergen. So those Clostridial organisms are protective against the peanut allergen. And recently we've identified a bacterial probiotic formulation with colleagues over in Italy, just a Lactobacillus rhamnosus GG, that when given to children very early in life, under the age of 3 months, can protect them from developing a milk allergen response, or a cow's milk allergy. And it works around 50% of the time. So we are now looking at ways of altering that formulation in order to try and improve its efficacy. We know that the microbiome can also affect depression. Bacteria in the gut produce neurotransmitters, as well as influencing the immune and endocrine system, which can alter neurotransmitter activity in the brain. The production and consumption of neurotransmitters by bacteria in the gut obviously alters the balance of neurotransmitters in the serum, which can affect neurological activity across the body. And we have seen many instances, and there's more work coming out, where we can alter the neurological or depression phenotype of an animal by affecting its microbiome. In one of the most elegant studies, which we say at the beginning, low-hanging fruit study, if we take a specific pathogen-free, a typical not germ-free mouse, typical mouse with lots of bacteria in it, we can see that the mouse will hide inside the boxes of an elevated maze. This is just a plus maze. It's a cross about 1 meter off the ground. And the standard activity of a mouse, standard behavior, would be to hide in that box and stay hidden. What we are interested in doing is looking at what happens when we take the bacteria away from that mouse. So when you make this mouse germ-free, it loses all that fear, loses all that anxiety. It no longer hides inside the box. It walks along the platform and explores its world as if it doesn't have a care in the world. In the natural world, that mouse would be eaten because it wouldn't stay hidden. And you might think that this response to stay hidden would be a genetic predeterminant, nothing to do with the microbiome. And that may be true, but the microbiome is shaping that response. So whereas the mouse that was brave and ran around got eaten, bacterial interaction with the immune system and with the brain and nervous system appears to affect the behavior of this animal in such a way as to lead to a improvement of that phenotype. So it stays within inside a hidden environment. Take all those bacteria away, the mouse goes crazy. So we need to understand that in order to be able to understand depression explicitly. Also know that bacteria can cause obesity. One of my colleagues was morbidly obese, 385 pounds. He lost 113 pounds by eating a traditional Chinese whole grain diet over about a 9-month period. And over that 9-month period, he lost a lot of weight, obviously, but he also lost a bacterium, Enterobacter B29. And when he cultured up that bacterium and added it back to mice, the mice put on a significant amount of weight. So in this instance, we have a bacterium that can, a single species, which can induce a phenotype in a germ-free animal. And that animal is eating the same amount of food as the other animal, but we think this bacterium is helping that animal to process that food more efficiently and make more energy available to the host. And also, we've shown later, it can also disrupt the hormonal system and liver functionality and disrupt circadian rhythms, which can lead to an increase in fat deposition due to a stress response. So where do we get our microbiome from? I don't think that video is going to pull that in the video. There should be a video there, but it doesn't matter. We get our microbiome mostly from the environment we find ourselves in. Obviously, imagine you are a child. I'll move on to another slide. Oh, there we are. Imagine you're a child growing up in an environment where, for example, you were exposed continuously to the Amish farm. You're going to be exposed to ducks and pigs and chickens and horses and cows and etc. And their microbiomes are going to mingle with yours to a certain extent, or at least influence your immune system to develop in an appropriate and structured way. But if you bring a child from the OR, from the C-section delivery, straight back into, say, an apartment on the 31st floor of a skyscraper in Chicago, then that child is not going to experience any of those other stimuli. It's only going to experience really bacteria that come from other people in that environment, because our houses, our offices are reasonably sterile. We know that the bacteria that come off of our bodies colonize these environments. There are around 36 million bacterial cells leaving your body every hour, and they colonize and coat everything. Most of the time they die, but we need to understand what that death or what that survival looks like in order to be able to better design homes and environments to make them more health promoting. So we've been examining this space and looking at seeing ways if we to map the microbial interactions between, say, people and their surfaces in their homes. So in this study, for example, we have 4 individuals, 2 parents and 2 children living in one home, and we tracked them daily over 6 weeks with their hands, their feet, and their noses, and then looked at the kitchen floor, the bedroom floor, and various surfaces the hands would normally interact with, in order to be able to map how the microbes were moving between those surfaces. The one thing we saw was that there was a more significant microbial interaction between the mother and the children than there was the father and the children, but there was also a significant interaction between surfaces that would normally interact together, so the feet and the floor, the hands, and surfaces that touch hands. You can see hand 1 is the hand of a young infant, and that his microbiome is significantly interacting with the bedroom floor. Let me just see if I can grab a laser pointer to point that out. So you can see hand 1 is interacting with the bedroom floor much more significantly down this loop here. And that interaction is because that was an infant crawling on the bedroom floor, and so his hand was more significantly microbially interacting with that space. This is important if we want to be able to track where people interact with environments and what kind of microbes they may be colonized by. We see that if we add dogs into this scenario, this is a family with 3 dogs, that the degree of microbial interactions increases exponentially. Dogs act as a microbial superhighway conduit, if you will, moving bacteria from the outdoors into the home, as well as moving bacteria more freely between people. Couples that have a dog are much more likely to be more microbially similar to each other than couples without a dog, because the dog increases the degree of microbial sharing between the couple and leads to an increase in microbial similarity. So we're very interested in how to understand and develop this structure. In fact, my wife made me get a dog on the basis of this research. His name is Captain Bo Diggly. That's a picture of him. And he has actually been increasing our microbial diversity of our home. So we have him to thank. I talked a little bit in the beginning about how your unique microbial fingerprint could be used for microbial forensics. So I thought I'd take a little detour out of health and just talk about this very briefly. Sorry, I need some tea. So, for example, here I have what I call a source tracker map, whereby I've got sources to the bacteria that are found on, say, if you look at the top right-hand corner, the kitchen counter. Just going to grab a freehand laser pointer. So, so for example, if we're just focusing in, so just focusing on this one. Here we have a kitchen counter, and I have 3 people and 3 dogs, if you look at the right-hand side. And I'm looking at how their microbiome contributes to this surface over time. If you look at person one is a young man at age 25 living with his parents. Person 2 is his father, and person 3 is his mother. And then there are 3 dogs. If you look at the time point I've highlighted on the kitchen counter under that red arrow, 100% of the bacteria found upon the kitchen counter over that time period come from the mother. In fact, the majority of the bacteria, nearly all time points, come from the mother for the kitchen counter. So this is obviously a very gender imbalanced environment. She actually goes away shortly after that, around here. Around here, she goes away and she leaves the home, and her microbiome starts to decrease in the kitchen counter, and the father's microbiome starts to increase. And this is because he's been left to his own devices, we assume, and he had to do the cooking, and therefore he microbially interacted with the kitchen in a much more robust way. So we can use the microbial fingerprints left behind our people in order to be able to understand that more effectively. One thing that's very interesting for us is that being able to track and map this could be used in criminal investigations. And indeed, we have been able to start exploring this. The face on the right-hand side where it says "deal with it" was the unfortunate cover of a Time magazine article highlighting this. And these photos are from Time magazine, so I apologize for the cover. But in this instance, what we are dealing with is working with the Chicago and the Florida PD, to the police department, to see if we can use the microbiome as a forensic tool. So we have mocked up break-ins around the country, and we send in our team. The young man in the bottom right-hand corner is actually that 25-year-old young man. But his name is Jared Hampton Marcel, and he's the team leader for this effort in our group. And he goes around the country working with local forensics departments to see if he can identify who broke into the house. Obviously, we know who broke into the house because we have the burglars in our team. So we see if we can identify it in different environments. And we're building up a database of some 2,000 individuals to see if we can improve the probability that we can identify an individual based on their microbiome signature. A little fun aside, but just to point out that the clinical research that we're doing is also having a significant role on other fields. Getting close to finishing up here. We know that the microbiome is very important in hospitals, so we wanted to take the kind of research that we did in homes and apply it to hospital environments. This is our new $800 million Center for Care and Discovery at the University of Chicago. It opened in February 2013, but we managed to get in there in January 2013 and explore it for around 2 months prior to the opening, when it was very, very clean and there were no patients and doctors. And then we continued to monitor it every day for 365 days, examining the patient's skin, the patient room samples like the floor and bed rail, the air and water samples, the hospital staff and the nursing station. We did this every day for 365 days to build up a picture of how the microbiome developed in the hospital. Bear in mind, we had a brand new hospital, and then to examine ways in which to, if that related to any clinical outcomes to the patients. So we're really looking at microbial sharing, but on a very grand scale. This image shows the floor microbiome as it went from being unoccupied to occupied. So the blue dots, the blue cluster of dots on the left-hand side are from an unoccupied hospital part. And then as the hospital became occupied, the dots move over here to the red. The occupied hospital is a very different microbial community. Obviously, the microbial community from the occupied hospital comes predominantly from the patients and the doctors and the nurses and the cleaning staff who are shedding their microbiome into those environments. In fact, we could identify which patient was in which room at any given time based on the microbial signature on the floor or on the bed rail or even on the wall because their microbiome was being shed into that environment. One thing to note in this was that the infections that people suffered, the healthcare associated infections that people suffered in that environment, we found no correlation between the presence of that infection in their clinical record and their microbial interaction with the environment. So we could find no evidence to support the fact that that bacteria came from the hospital. In fact, in nearly all of the instances, and we had quite a, well, not a significant number, but in the tens of the 400 patients we analysed over the year, of those 20, 25 patients, none of them, well, they all appeared that the microbiome, the bacteria that infected them, may have been present in their body when they came into the hospital. Either that or they acquired it as they walked in the door, which seems extraordinarily unlikely. And it was very different for each one of them. This suggests that we should start thinking about ways of examining the microbiome of a patient when they come in, and maybe thinking about ways of treating the patient when they're in the hospital. And not just treating the patient, but treating their microbiome as well. So obviously I'm in the Department of Surgery and I work very closely with surgeons. One of my colleagues, John Alberti, has been a significant driver towards helping me to get involved and understand the microbiome in surgical infection. So surgical infections are obviously a major problem. We understand them to be predicated upon the potential for surgeons or the environment to cause a bacteria to get into a site, colonize it and cause an infection. But in many instances, especially gastrointestinal surgery, there's no real evidence that the infective agent actually comes from the outside. And in some situations, such as gastrointestinal surgery, where we're producing anastomosis, for example, we resect a piece of gut to remove, say, a cancerous polyp region. We do two things there. We actually cut open the gut and pump in oxygen, which actually nullifies some of the bacteria, kills some of them, and stresses the rest. And we pump the body full of antibiotics, and those antibiotics also affect the health of the individual and the microbiome. Then we stitch up the gut, forming an anastomotic junction, and this is like a final insult. The stress the body's under starts to cause the body to sequester nutrients from the gut, causing a nutrient-depleted environment, which stresses the bacteria even further and causes them to change phenotype, producing a colonization of the wound site, and they release enzymes which start to break open the wound site in order to get nutrients back, and they cause leak. And in this instance, a leak, an anastomotic leak, can be fatal. We've identified bacteria present in animal models prior to surgery that then change phenotype due to the stress they're under and colonize that site. We think that phosphate and nitrate stress may be among the biggest drivers, and so we've actually identified particular chemicals that we can add back into the experience of the patient to help that patient to improve their health over time. So in this instance, we're actually able to significantly advance our preposition that surgical infection may be due to bacteria already present in the host, maybe on the skin, maybe in the mouth, maybe in the vaginal cavity or the urogenital cavity, or maybe even in the gut, and that those bacteria need to be kept, for want of a better word, happy or stress-free in order to prevent them from activating a virulence mechanism and infecting their host in a particular way. So for our perspective, it's very important to be able to understand how to treat the microbiome as well as treating the patient. And we are significantly altering many of our surgical practices in order to be able to do this. So just to finish up, I just want to highlight again our Microbiome Center. We are, this is a new initiative. Bear in mind, I'm an ecologist, but I'm also now working as a professor of surgery. And this is a paradigm shift that all medical campuses are really starting to embrace. The fact that ecology in the body is as important as understanding most clinical concerns seems axiomatic to me, but is a new paradigm for many clinical care programs. So the Microbiome Center actually brings together scientists studying the industrial application, marine and terrestrial environmental applications, agricultural applications, as well as health and animal and plant welfare, in order to be able to use all of that transdisciplinary expertise to improve and significantly advance the scientific program in each of the individual areas. One case in point in this, and it's an interesting one just to make, the research that I just showed you on surgical microbiome actually originated in part in understanding that bacteria in the ocean activate certain mechanisms to sequester and reacquire phosphorus from oceanic organic material when they enter into a phosphate depleted environment. So when there's not enough phosphate around, bacteria in the ocean activate mechanisms to get the phosphate back, right? And we went into patients, and when we saw that the phosphate in the gut went down, we went and said, well, maybe the same thing's happening in patients. And indeed, the same thing happens, the bacteria in the gut, when the phosphate goes down because the body is sucking it out of the gut to repair itself. When the phosphate goes down, the bacteria feel that stress and they activate a phosphate acquisition strategy, which is why they then colonize the wound site to break it open, degrade it down, and grab the phosphorus back. So by partnering up between seemingly extraordinarily disparate areas, we're able to significantly advance research paradigms in areas that wouldn't normally see the light of day. Thank you very much. It was a pleasure talking to you, and I'm happy to answer any questions.
Well, thank you very much for that amazing presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we possibly can. And our first question is, Dr. Gilbert, is it possible that the mental disorders are relationed with microbiome?
Right, yes, can you hear me? Yeah, okay. Yes. So, we actually, I work specifically with depression, especially perinatal and postnatal depression in women, neurological development in infants and children, and have a very particular interest in autism. In all of those instances, there is considerable evidence from our animal models that altering the microbiome can play a significant role in the development of those conditions. So, for example, if we look at autism, the work of Sarkis Mazmanian and the late Paul Patterson out at UCLA and Elaine Hassau has demonstrated that the bacterial communities inside the gut of an animal that is being activated towards an autism developmental phenotype can have the neurological symptoms of the disease significantly suppressed by altering the bacteria present in the gut. So by adding in a particular type of probiotic, they can actually suppress things like the stereotypical behaviors that those animals demonstrate, as well as some of the anxiety behaviors. There's new research coming out that shows that a microbiome transplant from a neurologically healthy rat to a rat demonstrating a Fragile X syndrome style autism behavior can affect its social structural behavior, so it becomes more socially cognizant. So there's a lot of evidence to suggest there's an interaction. The mechanism is still hard to pinpoint, but as I said, bacteria in the gut produce things like the precursors of serotonin, and they produce neuromodulators like GABA. They produce acetylcholine. All of these, some of these can pass the blood-brain barrier. Things like GABA seem to activate neurological receptors in the gut. Hormonal imbalances, they can bind up the GABA can bind onto hormone receptors, and they can also affect neurological transfer of impulses via the vagus nerve. In fact, in some studies, we've taken probiotic lactobacillus, added it into a mouse model, and the mouse will show altered GABA productivity in the brain just with the introduction of the probiotic. If you sever the vagal nerve, if you give the mouse a vagotomy, then the probiotic does not have any effect upon GABA activity in the brain. So there's definitely an interaction between the gut and the brain, right? Anyone that's ever felt happy or sated after a meal understands this. But we're starting to understand that it could also be playing a role in neurological disorders. And so there's a lot of work ongoing to try and understand those specific interactions.
Thank you very much. And our next question is, Doctor, do you have any data on microbiome during remission or recurrence of primary tumors? And do the type of oncological cures alter the microbiome?
Okay, so yes, there's some very interesting work, well, a number of interesting studies that should highlight on cancer in the microbiome. So I work very closely with groups up at the Mayo Clinic and various other communities here in Chicago, where we are very interested in trying to understand the particular types of bacteria that could induce rapid inflammation at the gastrointestinal wall site and therefore affect the development of precancerous polyps and then heighten the risk of developing a cancerous polyp. So from our instances, we've identified bacteria that can specifically increase the risk of developing a cancerous polyp and exacerbate that problem. There's also compelling work coming out of animal models from our groups here at Chicago that have demonstrated the potential to improve antibody-derived or driven antigen-activated cancer therapy, such as immune blockade therapy for melanoma. And we think that there's evidence from the animal models that we're able to significantly improve the efficacy of the treatment by adding certain bacteria back into the experiment. The prerequisite there is that, or the understanding there, the mechanism, is that the bacteria activate an immune response, leading to heightened potential for the white blood cells to reactivate and access the aberrant tissue. So, by using immune blockade therapy to target the cancerous tumor, you're actually able to increase the immune inflammatory response with the bacteria added into the experience of the animal, and therefore alter the efficacy of the therapy itself. So this is ongoing research, but there is definitely very specific links between oncology and the microbiome.
Thank you very much. And doctor, my question is this, can microbiome be the cause of cancer?
Well, in reference to the last question, well, not necessarily its only cause. It can play a role in the development of that, as I've just highlighted. The fact that bacterial can affect inflammatory responses, which can lead to a significant increase in the likelihood of developing a cancer if you're predisposed, is the current paradigm.
Thank you. And can you answer this? How long does it take for one person's microbiome to be replaced by another's in a defined room?
That's a really good question. So if the person is, well, it all depends on the context. But if I have one room, a controlled room, for example, and I have one person walk into that and spend, say, 10 minutes actively doing something in the room, walking around or lifting up objects, and then I have them leave that room, and then I have another person come into the room shortly afterwards, we would still be able to detect their microbiome of the first person for around 72 hours if that second person remained in the room for 72 hours. So for around 3 days, it's reasonably detectable, or at least above the probability frequencies. But if, for example, someone broke into a room and that room had 15 people living in it, or a house with 15 people living in it, and there was a massive amount of activity constantly going on, then it's likely that their microbiome signature would be eroded significantly faster and would probably become unidentifiable. That's the reason the microbiome for forensics research is not a replacement for anything. It's just a potential addition. It's another piece of trace evidence that, if applicable and if useful at a particular contextual system, could be used.
Thank you. And doctor, does the biome really influence microbiome and eventually our health?
Oh yeah. Okay, so does the phagome is the question. A phagome is related to viruses and phage, specifically phage that attack bacteria, or viruses that attack bacteria. Yeah, I think the phage is incredibly, or the phagomics is an incredibly exciting area. We have some very interesting data that could suggest that phage that attack bacteria when they become abundant in a particular area could actually be the trigger for certain disease phenotypes, even pulmonary diseases. So bacteria building up in the lungs themselves do not activate, say, a fibrotic event, a fibrosis. But the phage that then attack those bacteria, because those bacteria become abundant, the phage then become abundant because they are using the bacteria as a host. Those phage can activate an immune response. Certain toll-like receptors, such as TRL9, can respond to those phage. And once those toll-like receptors are activated, they can significantly elevate the likelihood of localized inflammation and then trigger a fibrotic event. So yes, the phagome is becoming an incredibly important element of helping us to understand how the microbiome is, what role the total microbiome, that's the bacteria, the fungi, the viruses in the body, and even the archaea, what role they're actually playing. We actually think that archaea are actually far more diverse than we previously thought. They're low in abundance, but they're very diverse inside the body. And so even the archaea could be playing a significant role in our health. This is still a very, very early phase of investigation.
Thank you. And do you foresee facial or nasal testing of the microbiome for patients pre-admission testing or prior to invasive procedures in the near future?
Yes, absolutely. So Patricia, you're very spot on. For us, especially, we're looking at low-hanging fruit at the moment. But for example, with pulmonary pneumonia, if we look at the oral microbiome of patients, if they're going to be aspirated, then we can actually see a correlation between the types of bacteria present in their oral cavity and the chance that they will get aspirated pneumonia. So we are very interested in that particular area, and for other treatments, such as even looking at their risk, the nasal and oral or oropharyngeal communities could be extremely important as a proxy for determining what's going on in the rest of the body. We're not there yet. We need much bigger databases, but it's definitely going to be something that we do pre-admission in the near future in order to reduce the likelihood of complications during treatment.
Thank you. Thank you. And what is the current understanding of the relationship between microbiome and allergies?
So, the basic preposition is that the microbiome plays a significant role in regulating the atopy or allergic response of an individual through a number of routes. Some bacteria are pro-inflammatory. They can release compounds which are recognized by the immune system and can lead to a significant increase in inflammation. Other organisms are anti-inflammatory or can reduce the inflammatory process by producing things like short-chain fatty acids, butyrate, propanoate, acetate, and butyrate specifically has a role to play in recruiting Treg cells, regulatory T cells, into the epithelium of the gastrointestinal tract, and those regulatory T cells produce compounds which suppress inflammation in the rest of the immune system. So that dance, or the interaction between the bacterial community and the immune inflammatory response, we think is the biggest driver for allergies in general, seasonal food, or anything else you're allergic to. So we are very interested in trying to understand that, and many of the commercial therapies that are being developed are looking at ways of increasing the types of bacteria inside the gut of infants especially, that are able to recruit Tregs and accumulate Tregs, regulatory T cells, in the gastrointestinal tract. So that's the basic premise right now.
Thank you. Are occupational diseases enhanced or limited by microbiomes? For example, solvents or heavy metals or various lung diseases?
Yeah, that's a good question. It's possible that heavy metal exposures and solvent exposures, and even say coal mine dust exposures could influence the microbiome, and that could be a component of the disease phenotype. You want to be careful about trying to hang too much on the microbiome, but the bacterial communities, archaeal, fungal, viral communities inside our body, they're all part of the system, right? And anything that perturbs them, or anything, that could be the body's own response to something perturbing or killing off certain types of bacteria, is going to have an effect of some kind. So it's important to be able to track that, understand it, and see if we can really start to piece together the role that this disturbance could play in a particular disease phenotype.
And thank you very much. And Dr. Gilbert, can a 2-week yearly vacation to developing countries change one's microbiome? I've noticed one change in my health after a trip to Thailand, where I spent a month. And how long does a person need to be exposed to a different microbiome environment to change their own microbiome?
It's a very, very complicated question. We don't know the answer. I mean, one of my colleagues, we had a paper out where they monitored their microbiome daily for 365 days, but they did it in 2 people. In the people that went abroad, in the person that went abroad from that 2 people study, their microbiome did change within days of being in that new environment. But only about 5 or 8% of the microbial community was affected. The rest of it remained relatively the same. And as soon as they returned to the US, their microbiome bounced back to its old self. So that's probably diet-induced variance, right? Understanding how diet or changes in diet because of your new environment could affect the microbiome is important. The important thing is if you are abroad and you get a traveler's diarrhea or another disease, that's going to affect your immune system, which is obviously going to affect your microbiome, and that could have long-term consequences. We know that that happens in many individuals that get those kind of diseases. So traveling abroad is going to have an effect, but the specifics of that effect are going to be very personal, independent.
And how advanced is the research in children under the age of 5 at the Microbiome Center, and does it include peanut?
So, well, if you mean for allergies, for food allergies, there's a significant increase in research of children, especially infants and toddlers. And so we are working on that right now. We are a food allergy research and education network hub, node. So we are increasing the research in that space. And there's a group of researchers at the university who are working very specifically in that area. And peanut allergy is one of the conditions being examined.
And what other diseases do you find interactions with microbiome, and can they be reversed by changing the microbiome?
Well, that's a very, very big question, not one I can answer right now. As I said, I listed probably some 30 or 35 different diseases that have a role to play. And as I also said, the microbiome is part of the system. So you change the microbiome, you're going to change some part of the system. So the interactions between there are incredibly complex.
All right, and we have time for one last question, so thank you very much, Doctor. And it's, do you think that I'm so sorry, it is in regards to weight loss and bacteria related to weight loss and how this would improve treatment on obesity with the microbiome.
So there's one study that's identified bacteria that can play a significant role in improving disease phenotype. So very specifically, that study looked at whether children who were lean versus their identical twin who was obese had enrichment of certain types of organisms, and they did. And when they try to isolate those organisms, add them to mice, it protects the mice from developing obesity. So there is work ongoing to look at weight loss or weight gain prevention in a specific field. So that's ongoing.
All right, thank you very much, Dr. Gilbert, for your presentation. And do you have any final comments before we wrap up?
I'd just like to say, there's a wonderful series of questions. Thank you very much. Bear in mind, the microbiome is not the panacea or be-all. It's just something that's been recently identified that could play a role in helping to improve disease and health treatments for particular individuals. So we are very interested in exploring it in a precision medicine capacity and in trying to use it to determine why some people respond and other people do not respond to treatments or have very variable symptoms for particular disease states. So I think there's a lot of work that needs to be done, but it's an exciting time.
All right, thank you very much. I would like to thank the doctor once again for all of his contributions and for his presentation. I'd also like to thank LabRoots for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through December the 7th. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.